Let me also tell you that when John joined DuPont from Princeton, he had just finished his PhD. You are not a big man then; you are just a starting researcher. The big man was their research director. DuPont, of course, is one of the great companies known for its fibers and polymer, all the great work that it has done. But DuPont was really a small player in the area of drugs. DuPont was never big in drugs. But because drugs is such a big thing, they didn't want to be completely left out. And so DuPont had created a small research team to work on this. And specifically, DuPont had formed a team for cardiovascular drugs. And around 1982, which was around the time that John got hired, maybe a couple of months after that, they had hired this person, Robert Tabor, who had vast experience at one of those famous pharma companies called Schering Corporation. And so they told him that, why don't you develop the cardiovascular research group in DuPont? He had come from Schering Corporation, which was itself a very big company, and he knew the workings of pharmaceutical companies and how big they are. He kind of looked at what work they were doing, and he said, how many of you are there? And he said, what's the point in doing all this work? There are so few of you, and you are doing more or less the same thing which everybody else is doing, which is, he felt that it was me too in nature. And he said, therefore, he put a stop to all that. And it says, Tabor decided to quickly put an end to these lines of cardiovascular research, which were me too in nature, with low chance that any of it would result in a drug. And then Tabor said, look, I mean, if you're a small group, work on something which very few people are working on, and it is high risk, of course, I know that, and maybe nothing else will come out of it. Maybe some good publications will come out of it. And he said that in any case, nothing will come out of the other approach. So you might as well go around along the path, which has been less trodden. That is what great people do. And then he said that this line of research was one that was less explored and believed to hold the highest promise of yielding something novel, if not a drug. And then John told me, no, that was not the only reason. Another reason was that although we talked about the ACE inhibitor being quite effective, but giving you a dry cough, but that's not the only thing. John clarified to me that it is always better to try and block a receptor than to block an enzyme. And so he said that, you'll get less side effects if you block a specific receptor. And you can see that we are talking about a specific receptor. That receptor is AT1, this receptor that you have to try and block. So that was the reason, and that Robert Tabor knew that. And so he said, look, let's just work on this. I hope you understand the role of a research director. A research director is like the conductor of a great symphony. And imagine everybody is playing their violin and this and that, saxophone, trumpet, but it is the research director that brings harmony, that helps to coordinate and convert something which is ordinary into something which is extraordinary. That's what a research director does and sets the direction. Okay, you are meant to set the direction. Okay, now, so let us try and see that at the time John Duncia got into this act, he was not the only one. DuPont had some brilliant people, a couple of people from MIT, John from Princeton, and a few other people, and really developed a great team. Now, what was done, as I told you, you see, this was A2, as we discussed, angiotensin 2. So there were companies who were making peptide mimics, short peptides, okay, which have a lot in common with A2, but are not exactly the same as A2. And both of them actually, the initial amino acid was sarcosine. And so that's why all these names are, because it starts with SAR, Saralacin and sarmacin. These were the two leads which people were working on. These are their structures. They resembled it, so they could fool the receptor into believing that A2 is binding. And they dislodged A2 and went and sat there, and they were harmless. They did not lead to vasoconstriction. Okay. But the problem was, as I told you, in the 80s, peptide related drugs were few and far between. People, as I told you, were working on simple, well defined organic molecules. And so John says, we could not discover a small potent peptide fragment of A2 or saralacin, which we could then analog into a small molecule drug. Because that's what he was comfortable with. He's an organic chemist. He's comfortable making organic molecules. And, so that's what he set out to do. Okay. Now, but this is the other thing. There is always somebody who has done something remarkable. And I asked myself, did DuPont really do something so great? They, of course, became very famous, and John became very famous. But then I realized, no, the people who did the very original great work were again the Japanese. And this company called Takeda, around that time when these people had joined, you can see the date, October 19, 1982, they reported the first organic molecule, which they claim is also an antagonistic action for A2, and it is a non-peptide. It's not a peptide. Okay, it's a simple organic molecule. Okay, and they reported it, and subsequently they followed it up with two more patents, and probably thinking that they had really covered it very well. Their patent, and you can see they started their work well before DuPont. Filed in November 1980, which means they must have been working on it from probably 1976 or 77, thereabouts. And so long before DuPont. Now, and they covered here, you can see the main thing, what they claimed was basically an imidazole derivative. And this is what it was, CH2CO2R. And there were some substituents here, R1 and Y. And then they had an aromatic ring of a benzylic moiety of the nitrogen. And here again, they claimed some substituent. And so they were quite thorough, or so they thought. And so, that's what it. Now, what DuPont did was that they read this patent, and very quickly they made a few molecules bearing this kind of a structure. And I've given you their code names. This is what they made. As you can see, it's identical, because some of the X1 and X2 were actually, they've said it can be H. So H was also part of X1 and X2. So quite similar. And you can see again CH2CO2H, same. There's a Y, so they had put a Cl, and there's an R group. So they put an R group, I think a butyl group over here. So what they did was they didn't make only one molecule, they made several. The X, which was coming off the aromatic ring, okay, this X, okay, that is the one where they put a NO2, Cl and H, and this was Cl, already claimed by Takeda. And they confirmed that there is an activity, a cardiovascular activity which these drugs show. But what they found is that it was 4 orders less active than those polypeptides, that saralacin and sarmacin. Okay, and so if you needed something like a 10 to the power minus 8 molar concentration of the polypeptide, this drug you needed 10 to the power minus 4 molar, 4 orders of magnitude. So of course it was a lead, it was great, but I mean it was nowhere near a drug, and probably that's why Takeda did not take it forward that swiftly. Maybe it was good for publishing a patent, but not really good enough to make a product out of. But the interesting thing was that they did a study with S8307, that is the Cl derivative. By the way, these are the IC50 data. That means the concentration at which 50% activity is seen. And so if I can get, for example, a 50% inhibition of antagonistic activity, that will be IC50. Okay. And so among these, the NO2 had the lowest concentration. That means it was the most active. This was the least active, and the Cl was in between. Whenever I see NO2, I keep thinking about the original work on nitroglycerin, and who knows whether some formation of N oxide might also be taking place. Anyway, but they did a lot of the work with the Cl derivative over here, and the remarkable finding, which was made by this person, Pancras Wong, again, he was hired from one of these very top universities. He was a pharmacist, okay? And what Wong found is that this compound is not very active. Of course, they had shown that. But the great thing was that it had very low toxicity, and he could take large amounts, 100 milligram per kilogram, which is you can imagine what kind of weight, say a person who is say 800 kg weight, obviously I'll have to consume very, very large quantities of the active drug, which is not practical. And but the fantastic thing was they found that it is very selective and very low toxicity. So they said, we must try and improve over this. Not very different from the way James Black and Stephenson went about improving their particular molecule. Tabor was happy because they had a lead, and he said, let's play around it. And as I told you, the degrees of freedom are quite high because there are so many substituents and you can do so many different things. Okay. Now, this is where John, I think, went slightly differently. And he could do that because already in the 70s and early 80s, NMR of biological systems was becoming very well studied. And they actually, John was able to actually find some of the conformations in solution of angiotensin 2. And so he developed a 3D mapping of this A2 molecule. Okay. And what he then looked at was this particular Takeda compound, S8307. And somehow John concluded that it seems that it is missing a carboxylic acid group. And he felt that if he can introduce a carboxylic acid group into this molecule, that maybe it will be able to compete with A2, and it will be a better mimic than what Takeda had found. That's interesting. And he says there were a few conformations for A2 in the literature, and I just naively picked one that I thought had good NMR work associated with it. Again, a bit of a hand-waving argument. He just picked one of the conformations, and kind of made this very, very interesting conclusion that it seems to be missing a carboxylic acid group. Now, then what he did was that they actually introduced a carboxylic acid group. They took out the chloride group, the Cl. In its place, they introduced a carboxylic acid group. Okay, and there were two kinds of interactions which these molecules had with the receptor. One was basically the fitting type of interaction, it's like a lock and key kind of fit. The second is that there were van der Waals and electrostatic interactions, and I'll show you that a bit later. Okay, and When they made this molecule, what happened was that he found that the activity of this compound was 10 times more than S8307. Out of the 10,000 times lower activity, at least they had made up 10 times by putting in the CO2H group. And John would like me to believe that he actually designed this. He designed it based on his understanding of the conformations. That's remarkable. I would like to believe that what he's saying is correct. I'm not so sure, but let me give him the benefit of the doubt. you know. Okay. The other thing that you have to do is whenever you have these leads, you have to make sure that it is showing a concentration dependence. Many times, there are artifacts. You get something and then you increase the concentration, but the activity doesn't go up. If the activity is not dependent on concentration, then it might be something spurious. So they did an extremely careful study, again, Pancras Wong, and what they found is that as they increase the concentration of this compound, the suppression of the arterial pressure became better and better. And the moment they were injecting the rat with this drug, this stuff, immediately there was a decrease. Okay, so over here, you can see they introduced it and it dropped. Okay, and after some time again it rose, again they injected it here, again it dropped, and because they were injecting higher and higher concentrations, so it was getting more and more effective. So that was the first lead where they got a 10 times improvement in the activity. Now, that was not the only thing that they got this 10 times activity. There was another remarkable thing that happened. And I asked him that I wanted to ask you how you dealt with the threat of infringement of the Takeda patents, considering that Losartan 2 is an imidazole-based derivative. I mean, Losartan is the final drug. We haven't come to that yet. And he sent me this first page, okay? And he wrote, they did not claim CO2H or any acidic groups of the benzyl phenyl. Thus, we had new art. And then he makes this in bold and he puts over here no CO2H. Now, what a tragedy for Takeda. They would have thought that they are covering themselves very well. Little that they knew that they have not included a carboxylic acid group. Who thinks of so many substituents? There's no end to the number of substituents. Okay, and so that was most tragic. Very, very tragic for Takeda, but very great for DuPont. So the CO2H, the introduction of the CO2H group did two things. One, it increased the activity 10 times, which gave them more of a kick, a real excitement. And the second is they got out of the Takeda patent and they could file their own patent. And so remember this, many times when you're filing a patent, you might be giving away more than what you're getting. And clearly Takeda gave away far more than anything that they got. And the people who gained were really DuPont. Okay, now then, what happened was that, he kept on working, all right, on this molecule. And what he said is that it's the molecule is still not long enough. The receptor is quite large and the molecule is fitting well, but is not covering the entire receptor. And so John felt that he must try and increase the length of the molecule. Okay. And the way he did that was that, he took this aromatic ring, okay, and there was an amino group here, and he basically coupled another aromatic ring, into this, basically forming an intermediate amide. Actually, it wasn't so simple because they made many molecules, okay, not just one, okay. But what happened was that, and they of course stuck to their CO2H group, so they had a carboxylic acid group still, and they made the length in the molecule by this. Now, Again, as luck would have it, when they made this particular compound, the activity went up 100 times with respect to S8307. So, they had only 100 more to cover, two orders of magnitude. That they had one, two orders of magnitude, and two orders of magnitude was still to cover. So, you can see how John and his colleagues played with the degrees of freedom which were available to them. The degrees of freedom which were also offered by the patent filing. Now, and they actually then drew the receptor. This is the pocket, okay, and this is how that molecule was fitting into the receptor beautifully, okay. And I mean, almost beautifully. It was not so good, you'll find out. Okay. And what they were able to show that the apart from fitting well, it looked like that there was van der Waals interaction. There was a kind of, not van der Waals, a hydrogen bonding interaction that was taking place here at one end, and at the other end, because of the carboxylic acid group at this position, in the ionized form, it was able to have a coulombic interaction with the receptor which was positively charged in this area. Okay, so it was held quite firmly in the receptor, and that's how they were able to keep out the A2. Otherwise, something can fit, but it may loosen and come out. Okay, but that didn't happen here. Okay, so this was really wonderful. They had their 100 times. Now, till that time, all the work that they did was on intravenous application. So they were injecting the molecule into rats. Okay. Now, someone would have told them that, look, atenolol, etc., are drugs that you can take orally. You take a tablet and you basically take it into your stomach. And intravenous is not something that people will be so happy about. Although, of course, people take insulin injections and things like that, it's not something that is so nice, compared to being able to just gulp down a drug. Okay. So at that time, their focus shifted to how to make the drug orally active so that it could be taken from the mouth. Okay. And, they played around. And again, let me tell you, this is all part of fortuitous work. Okay. Huge accidental luck. It is, of course, brilliance. I mean, I'm not saying more than brilliance, tenacity, to make so many molecules, test them out, etc. So what they did was they started playing with this carboxylic acid group over here, which was in an ester form. Okay. And what they found was that when they converted this into a CH2OH, a hydroxymethyl group, instead of this group. Okay. And they also subsequently knocked off, this amide which was there, and now they had a direct biphenyl ring. Okay, so this was biphenyl, and they kept their CO2H. Not very different from Knoll, where at all times he kept his anisole and cyclohexane. So here again, they were able to keep the CO2H group, probably because of the electrostatic interaction. And with this master stroke, they converted an injectable drug into something which was basically orally active. Okay. And so it says attachment of hydroxymethyl group to the fifth position on the imidazole ring made the molecule orally active. And so one after the other, they started getting away from Takeda's IPR, and they had that proof with them that they had made something better. It was not a fake thing. So they had 100 times higher activity, and they also were able to make it orally active. So quite a dramatic improvement. Okay. Now at that time, John did something else, and I would say that this is the turning point. The turning point, almost like osmium catalyst for ammonia synthesis. Everything is great, but the discovery of osmium was the turning point. Okay, so let me see what was John's eureka moment and turning point. Okay? And by the way, this all this happened when we were roommates, and he was a student. He would go to Barnes and Nobles, which was a very famous bookshop in Manhattan, and many times he would go on a Sunday, and you can go and they have these books they keep on the rack, and you can buy almost a sack full of books for a couple of dollars. And so he would pick up a lot of books. And John used to be more serious than me, he used to study hard, unlike me. And so, he picked up all this, and he had this book with him. He didn't read it at that time. Okay, but he had bought this book. And then what happened was that one day, when he was in DuPont, he started reading this book. He remembered about this book. He remembered about this book, and he remembered something about what are known as isosteres. Okay, I'll explain to you what is an isostere. And what John felt all the time was that he needs to make the molecule bigger, and he wasn't sure how exactly to make the molecule bigger. And the breakthrough that he had was, he asked himself, is there something like a carboxylic acid, an acidic compound, which is much more bulky than carboxylic acid group? Okay, that was the question that he asked himself. And he remembered from this book, and that's why he's put it down. Okay, he remembered that there are these compounds, the tetrazoles. Okay, and tetrazoles, if once they get ionized and it becomes like a CO2 minus, similarly a negative charge, then the system becomes aromatic. So it has a fantastic aromatic stabilization energy, and that makes it very acidic. Okay, the tetrazoles. All right, and that is what is known as an isostere. So tetrazole is an isostere of carboxylic acid group, something like the way Stephenson replaced two of those chlorides and put in a naphthyl group instead. So not very different. And so this is what John says, and I'll show you what happened when you put that in. Replacing a carboxylic acid group in EXP7711 with a tetrazole acid functionality. So this is your EXP7711. Mind you, and look at this also, that when the CO2H was in this position, it was orally active. When the CO2OH was in this position, it was not orally active. Okay, so this is the one that they chose, and they replaced the CO2H with this tetrazole group. Okay, and obviously this is a much more bulky compound than that. Now, replacing a carboxylic acid group in EXP7711 with a tetrazole acid functionality, the tetrazole improved oral absorption and increased potency. The activity was now 1,000 times more than S8307. So he got another tenfold increase in activity. And this is what was christened as Losartan, DUP753. And then John says something which I hope all of you will remember. Sometimes you have to take leaps of faith. Sometimes you are not lucky, but sometimes you get that tenfold kick in activity. That's what happened to him. And if you are very conservative, by sticking very close to the initial structural lead and are afraid to step out, you probably won't get there. Okay? Now, this was something dramatic, to conceive and envision a tetrazole as a mimic for CO2H. And now he says, nowadays, making a tetrazole as a CO2H isostere is a no-brainer. Back then, there were many in my department who never heard of this heterocycle. And even some thought it might be explosive. And so they were actually preventing John from synthesizing it. They said, "Are you crazy? It'll be explosive with 4 nitrogens." You know? And John had to persuade them to allow him to make this molecule in milligram quantities so that it could be tested out. Is it all clear till now? Has everyone understood?
Yes, sir.
When John, tried to model this and put it in the along with the receptor, what he found is that it has got a very good fit, and because the tetrazole moiety is quite big, probably the coulombic interaction that you can have with the receptor positive charge is better, stronger, and maybe that is the reason why the activity has gone up, because activity finally depends on what fraction of the molecules that you are ingesting, what fraction of that is actually binding to the receptors. Okay, that's really very important. So presumably that's what happened in this case, and this is what led to a thousand-fold increase in the activity over the S8307, which was the original lead taken from the Takeda molecules which were covered. All right. So is this clear? Have all of you understood? Okay.
Yes, sir.
Now, you remember that saralasin and sarmesin, there was a 10 to the power 4 times difference in activity. So we have covered 1000, but we still have another factor of 10 to go if this molecule is to really compete with sarmesin saralasin, because otherwise a tenfold higher concentration requirement is too high. Okay. Now, this is where again DuPont struck lucky. Before that, let me also tell you that after DuPont had discovered Losartan, and then, of course, when their patents ran out, there were many Me Too compounds, which a wide variety of companies actually started introducing. And you can see some of them, of course, were based on imidazole. These are all based on imidazole. This is not based on imidazole. This is a different heterocycle. Okay. And so what was very interesting was, doesn't matter what was the heterocycle, even if it was not an imidazole, losartan is this. Okay. In all cases, this part was more or less kept identical, in each of these drugs. And it's shown with this wiggly line. So beyond that, this part is really this. It's quite amazing that even if you change this imidazole part, this part was very, very sensitive. And obviously that made me wonder a bit. And I wrote to John, and I said, John, I just had a stupid question. Was a control experiment carried out to ascertain if tetrazole itself possesses any antagonist activity? I know it is highly implausible, but I thought I will check. Okay, and the reason is because I naively thought that that might be playing a important role. Who knows? That might be the active. Okay, and then he wrote back saying that we didn't screen tetrazole per se, but I believe we did screen the simple methyl biphenyl tetrazole, which means they screened this part, which makes sense because this is the overall substituent for these. Okay, and he says, and I don't recall it having any binding potency, at least in the 10 micromolar range. Then he says something very, very important, and that's the reason why I'm showing you this. By the way, there are no stupid questions. We always have another set of eyes not working on the project to review our progress. Since when one is too close to one's project, he or she might miss something obvious. And then he says, thanks for the great question. I wish you were with me on this project. What I'm trying to say is, I see a lot of inhibition in people to ask naive questions, and don't feel bad about it. There's nothing to feel embarrassed. Okay. I mean, if something is irking you or bothering you in your mind, all right, just ask. Okay, I didn't worry about it being a stupid question, and I asked such stupid questions in many cases, and it doesn't bother me at all, because it's troubling me. So I will ask. Is that clear? And I hope that all of you learn this very important lesson. Okay. Now, I said that we still have to cover another factor of 10, and DuPont were really lucky. What happened was, you remember that this CH2OH group, okay, earlier it used to be a CO2ET group, and this CH2OH group was introduced to make the drug orally active. So that was a huge thing. But what happens is that in the body, this thing converts, it gets oxidized to CO2H. Okay, CH2OH becomes CO2H. And this has 10 times more activity than this. Many times, if you're not careful, you might miss out some great thing that is happening in the body itself. Yeah. It's not that the drug that you're ingesting and that activity is the ultimate. Okay. So, what it means is that you need to understand the metabolism of a drug in the body. Okay. Look at the kinetics, look at all kinds of things. And that's where chemical engineers, especially, can play a huge role, because you understand these things. Okay. So that would be very nice. So I'll just read out pharmacological studies in rats revealing that an active, highly potent metabolite was created, as a result of which the overall gain in activity was 10,000 times that of S8307. This is where we started. And how step by step there was a 10 to the power overall increase in activity. And then it says something very interesting. The further finding that the metabolite tended to remain stuck for a long time to the angiotensin 2 receptors, much longer than Losartan itself. Okay, so this is the drug that you actually consume, but it converts to this, and this sticks around in the receptor longer. Okay, because of which enabling Losartan to remain active even when taken only once daily. Now, this is the kind of thing. Can you imagine what massive convenience there is to the actual people who consume these drugs? I mean, it's not the same thing if I have to consume a drug 3 or 4 times a day, and if I have to do it just once a day. Okay. And also the load of the compound in the body, because the liver has to metabolize all these things. Okay. Otherwise it will build up in the body. So these are so important. So what I want to tell you is that this is not really the what was the ultimate. It actually transformed into this. Okay, but this is the one which was known as Losartan, the very famous thing. And because the carboxylic acid group, once it ionizes and it becomes negative, okay, they tended to isolate it as the potassium salt. So you don't consume actually the acid form. This ionizes, it forms the salt, and that is what DUP 753 K plus salt. Okay, so this is getting replaced with K plus, N minus K plus. And that's really what is getting. Let me tell you that when you want to do really good drug discovery research, and that's where I believe that you require people who are good at quantifying. And so all these numbers had to be quantified. So, for example, oral bioavailability is 33%, it's not 100%. All of the molecules were not bioavailable. Two-thirds was actually just getting ejected from the body. And then 14% converted into EXP3174, but it must have been so active, this EXP3174, this with the CO2 age group, that overall, you got a very, very high rise in activity in the body. And after you consume a drug, it may take some time to get the peak activity. Okay, so the T max, the maximum activity is shown with Losartan after 1 hour, whereas with EXP3174, it is in the range of 3 to 4 hours, and the half life, of the Losartan is about 2 hours, and this is about 6 to 9 hours, the half life. So, very, very significant changes which happened because of oxidation. And another very important thing which was found, and extremely important, that no evidence of accumulation after multiple dosing. This is again very important, because if there is accumulation, then toxicity will build up in the body, and you will not know how much to actually consume in a day, because it's continuously building up. So this was also vitally important, and all this data is has to be generated, and then only you can say that you have got a safe drug. Now, John has also, I asked him, because just like William Knowles, I asked him that can you put down whether there were any fortuitous outcomes, things which you didn't actually plan for, but it happened. And those were very beneficial in your drug discovery process. And he says that the first is that when they were getting started, the Takeda patents had just been published. So they synthesized the right compounds, and you saw S8307 was the right compound, because what they found is that it was extremely safe. There was very low toxicity, and that was a huge motivation. Now, some of them are too technical, so I will skip those. Okay, but I'll come to this. Well, one thing was, you remember that we said that it is the 81, okay, the receptor which they were targeting, because that is the one which was lowering the blood pressure. If you could find an antagonist for that, and the way in which they designed their assay was targeting the 81, and so they could understand the selective binding of Losartan to 81, because there's 81, 82, and they were not concerned about 82. So that's what they showed with the kind of assay that they developed. Then he says that 6 assumptions were used to align the Takeda lead onto a solution conformer of A2. Now, that is also a luck that the conformer that he chose turned out well, because there were many other conformations reported for A2, and the one that they chose seems to have worked well for them. And then they made certain assumptions in how exactly the molecule will align in that receptor, and you remember that that is what gave him the clue that it is missing a carboxylic acid group, and that's how the whole thing got started. Okay, and that's what he says. The model worked because of the presence of 2 carboxylic acid groups, which increase the activity 10 times. Then the other luck for them was that the carboxylic acid group, which turned out to be so important, that group is the one that Takeda did not claim, as one of the substituents of the aromatic ring. So this is what enabled DuPont to actually patent their work. Otherwise, nobody would have allowed them to continue with this, because if they were infringing on Takeda's patent, obviously there would be huge risks. Okay, and so that miraculously, or whatever, that Takeda did not cover turned out very well for them. Now, although we today take it as for granted, but tetrazole, because it has got 4 nitrogens, people thought that it will be an explosive molecule. And so there was a lot of inhibition in their lab to actually allow the preparation of tetrazole and substituting it, because people thought it might cause an explosion. And I had mentioned this before. Okay. And they were lucky that there was no explosion. And thus he says that the first synthesis of Cozaar did not lead to any explosion. That allowed him to continue with his work. Otherwise, he may have had to discontinue it. Okay. Then this was again fortuitous that the CH2OH group oxidized in the body and increased the potency 10 times. That was a huge thing. And what he says is that the fact that the oxidation is not an instantaneous process, it takes time. And so what happened was that overall, they got a very smooth profile for the onset of action. I mean, it was almost like as if there is a zero order thing. So molecules were being produced, gradually molecules are also getting consumed because of metabolism. But that's gave a very nice, smooth onset of action. These are, of course, things that pharmacologists understand well. Now, another very, very interesting thing is that this oxidation to CO2H worked well or was seen to happen in rats and human beings, but not in dogs. And they were lucky because obviously they were working with a rat model. And so all their work was originally done with mice and rats. And so it was fantastic that it was there. And you'll also understand that not that, everything will necessarily give this oxidation. Like, as I said, it doesn't work in dogs. So, very, very interesting things about these kinds of drugs, and and that's why he says that we were fortunate that the blood pressure lowering effect in rats translated to humans. That's sheer luck. Okay. And then he says the longer T half of EXP3174 together with its insurmountable binding profile most likely accounts for Cozaar's long duration action. So long T half and plus that it binds there for a very attractive period of time. Okay. Now, after they've done all this, okay, when they wanted to commercialize this drug, remember, till this point, it is all drug discovery. Okay. And then they went to the DuPont pharma marketing team and they said, oh no, we are not interested in this molecule. And they said, why? They said, we have done a evaluation and we see that the market opportunity is no more than 20 million dollars, US dollars. Okay. And this, what was the reason? They said that they believe that the anti-hypertensive market was already saturated, being dominated by ACE inhibitors, captopril and enalapril. Okay, this was Merck, this was Squibb, and they wanted to stop the development of Cozaar. Let me tell you, and I have seen this in my own company where I worked in ICI many times. The biggest obstacle comes not from outside, it comes from inside. There are people inside an organization who will sabotage the activities. They don't want to put in all the work behind introducing a new drug. So they will cook up all these stories. And look, anyone could have been demotivated. But what John and the team did was something remarkable. They said, okay, if you are not interested, Mr. DuPont, can we go and now talk to other companies and see if we can sell the technology? And what better than to contact somebody like Merck, which was already into ACE inhibitors. So there was a drug. So any company which is already selling a drug, and if it sees a good opportunity for a superior drug, naturally they will be interested. Okay. And it's remarkable that Merck saw the opportunity which DuPont did not. And they said, let us do one thing that we will merge our activities and we will do a joint development and try to introduce Losartan in the market. Losartan was called as the Cozaar. I don't know who gave the name. And so this, he says over here, we were fortunate that Merck entered into a co-development agreement with DuPont to make sure Cozaar hit the marketplace. Now look at this. $20 million is what DuPont Pharma had said that it might sell. And within, I think, a couple of, or not a couple of years, a few years after its introduction, in 2005, the sales were $3 billion. And today, I don't know what the sale is, although it is not Losartan necessarily. I showed you that many companies have introduced these kinds of drugs. And I was talking to my brother, who's a cardiologist in Apollo, and he told me that there are better molecules now than Losartan. Losartan is considered as first generation. There are second, third generation drugs which are supposed to be even better, but doesn't matter. The foundation was laid by Losartan. So it might be touching, I don't know, 20, 25, $30 billion. That could be the kind of figure. So maybe about 1,000 times difference between DuPont's assessment and what is the reality. Why did it take off? Why is it that even in 2005 it sold $3 billion? And I told you, we discussed about Atenolol. This is the reason. In a 5-year, 9,000 patient study versus the beta blocker Atenolol. Remember, why did they do it versus Atenolol? Because Atenolol was the benchmark. There was nothing better than Atenolol till that time. Okay. But Cozaar reduced stroke by 25%. The risk of cardiovascular related death, heart attack and stroke, was reduced 13%. And in addition, John says, the chances of dying from any cause for diabetics was reduced by 39%. Okay, so it's just incredible the advantages that they got, and besides the advantages over things like in ACE inhibitors, the dry cough and stuff like that that I mentioned about. Otherwise, Atenolol is a very safe drug, no side effects, wonderful drug, but it could not match up to the kind of improvements that one got after introduction of Cozaar. For those who like process, I'll just mention to you how it is made. Okay, you take the imidazole derivative and you take the benzyl parabromobenzyl bromide. This molecule, by the way, ICT has done a lot of work on this molecule, and it's basically of something that we had made before, but we can now carry out this with 100% bromine atom efficiency, making this thing starting from parabromotoluene and introducing this BR group. So it could be a potentially lucrative opportunity if we were to go forward with that. So then you get this, it links up via this. So it is the HBr that is coming out and you're linking it here. And then what you do is that you take the other part, which is the tetrazole based part, and carry out what is known as Suzuki coupling. This is a palladium catalyzed coupling, and that's how you introduce the tetrazole group. I presume this will be common to all of the drugs, but this is the way Merck and DuPont were doing it, and you can see in 1994, long time ago, and this is now the world's largest Suzuki coupling. Suzuki coupling has become a very, very important methodology in synthetic organic chemistry. And the largest of that is for basically manufacture of this Losartan type of molecules. So again, there is a huge opportunity, and this is what India is quite good at, which is improving processes once a molecule is there. We are really not that good in discovering a drug. There are very, very few drugs which have been discovered in India, but we have been strong at the process end, and that's why for a long time in India, India only would recognize process patents and not product patents. And later on, many of these multinationals, etc., said it's ridiculous. I mean, the amount of money that we invest in kind of discovering a drug, and you say that you'll just make it by another route and bypass our patent, that's not acceptable. And that's how India was compelled to actually also accept product patents. So today in India, you can file for both product patents as well as process patents. Also remember, just like these fortuitous things that I talked about, but in the on road to making the cardiovascular drug, okay, they were very clear. They wanted a heart drug, okay. Now, this company, Pfizer, the same Pfizer which is now become big for the vaccine, Pfizer is a very big company. So they were also trying to make cardiovascular drugs, not the Losartan type, but some other type. And in the process, they had made this particular molecule, which they found to have good cardiovascular activity. That means it was also pretty good at reducing blood pressure and things like that. But the accidental discovery was, we have learnt about receptors, okay, and that receptors can be present in different organs of the body. This went and sat on the particular organs which control the erectile function in men, okay, and that's how this very very famous drug sildenafil was invented for people who suffer from potency, and it was purely an accidental discovery out of cardiovascular research. So remember this, and smart people, good companies, they will have multi-level screening. If I made a molecule, you don't just necessarily test it in one thing, you test it out in various things, and you might strike lucky. So just keep this in mind. I also thought I should mention to you that in most other cases, when humans make something, we call it an invention. In the case of drugs, typically the name discovery has remained. And you talk about drug discovery, you don't say drug invention. Okay. And I believe that is because all the original drugs were based on natural products. So it was a case of discovering these natural products, which will basically give you some effect of suppressing headache or this, that, whatever it is. That is probably the reason why the term discovery has remained. It has not gone away. And that's the reason why we probably still talk about drug discovery. We don't use the term drug invention. Okay. So I just thought I'll, just as an aside, mention this. Okay. Now we have covered and completed the drug discovery. Let me ask you. I'll just take 2 minutes. Is it all clear? Does anyone have any question? Did you understand everything?
Yes, sir.
Yes, sir. I want to ask one thing about the discovery part, which
Yeah.
you said. So these molecules, like, are they also naturally available, which can be synthesized in the labs? So,
Oh, many are, but not these.
oh, okay.